All right. Before we start, well, we can start now. But before we start with the course two, I can give a little summary on a paper I found, which was an article from 2016 that studied over 80 surfaces per kitchen, and from those samples, for of the 332 original samples that was analyzed, 248 were like useful, where like they found 34 bacterias and two archaeal phyla in the and the overwhelming majority of sequences, which means 98% belonging to four bacterial phyla. So you have the actinobacteria, Bacteroides, or Bacteroidetes, Firmicutes, and Proteobacteria. So those four bacteria are supposed to be found in all kitchens. And if I could have like a real petri dish and the DIY showing sort of the same result, it would mean that I did my experiment thoroughly. But I don't expect that, of course. I expect the DIY to be way more contaminated than the real petri dish. I'll leave that for another time. For now, we're gonna continue with the course too. So yeah, amino acids and hydrocarbons were identified with the Miller experiment. So when I mentioned it last time, I talked about like a circular model where you have uh, very raw elements. So methane, ammonium, hydrogen gas, and so on that were thought to be in the very early state of the Earth, and by basically zapping them, you could from the sample you could basically analyze that amino acids were formed, and longer chain of hydrocarbons were also formed. So what is putting things together and breaking them apart? That's basically dehydration and hydrolysis, typically in organic molecules. We can also call them metabolic and catabolic mechanism or pathways. Metabolic means that you build up. Catabolic means that you break down. And hydrolysis, for example, comes from the Greek hydro, water, and lysis means break. And dehydration, it's basically you remove a water. But as you can see, it's also
I'm
counterintuitive a bit. Yeah?
I'm not in a position to do this at this red hot second, but if somebody would take a few notes on these words and throw them into the Greek and Latin channels so that I can put them on Monday's slides, that would be great.
Yeah. Yep. So as you can see, you will actually add a water to break down a component, and you will also remove a water from polymerizing or from metabolizing an organic compound. Any question on either the Miller experiment or I'm not going to go in too many details in the Miller experiment. It's you feel free to read it, read the slide, and even look it up on the internet. But I just promised to show a little model of the Miller experiment. Any questions? Okay. Next, yeah. So we have sugars that may either be aldose or ketose, depending on the location of the carbonyl group, and classified according to the length of their carbon backbone. The spatial arrangement ar- around asymmetric carbons is known as the third point of variation. Chemical equilibrium between linear and ring formation favors the formation of rings. So as long as you as soon as you got the right carbon chain with the right conformation, propose to form a sugar, it will actually encapsulate itself and dehydrate, which means that it spits out a sugar, a water molecule, and is already a monosaccharide. Yeah. Any question on that?
So it's important for you guys to know that this is just like stuff to know that's out there, as opposed to suddenly being like, oh wow, now I need to learn a whole shit ton of organic chemistry. Awesome. Not necessarily like the whole, and then I decided to go down a rabbit hole thing actually doesn't will break your brain if you try to do that with OChem or certain types of bio. So it's more about
Absolutely.
having
Yeah.
having a general understanding that these things exist, and not actually being like, okay, now I'm going to learn how to make carbon rings, because
Exactly. Yeah.
don't do that to yourself.
That is exactly why I'm picking certain examples that I think is kind of peculiar, and I am going, like, I'm skipping many steps. I'm gonna talk about it later on, but I am basically showing what could be focused on to get a bigger picture, but also just yeah, showing that it exists, and one could, take a glance at it and think, oh, okay, well, it spits out a water molecule when it's building up a compound, or it needs a water molecule to break down, for example, in yeah, organic molecules. It can be important, but it's not only with sugars. It's going to be with many things. So yeah, we can go ahead with next slide, unless there is question or before. All right, let's go. Storage of polysaccharides in plant and animals. So you have starch, the plant polysaccharide that are found in two forms: unbranched amylose and branched amylopectin. The light ovals and are granules of starch within the chloroplast. And you have the glycogen, the animal polysaccharide, which is the more it's more branched than amylopectin, and stockpiled glycogen contain it as those dense black clusters where the mitochondria will basically knock onto that and break it down. Uh, Questions before next slide?
Are these just broken straight down into the different base pairs for DNA and RNA?
Yeah, it's a little introduction to that, indeed. Yep. Yeah. Next slide.
They're broken down into their constituent parts, and then the constituent parts are used to make stuff.
Yeah.
It's literally like taking these, you know, uh long molecular chains, imagine them like a brick wall, and then you're like, okay, I'm going to take this brick wall and salvage all of the bricks, and then I'm going to rebuild something else with it.
Right.
So yeah, like when you're talking about base pairs and stuff, these are still like, those are aminos and stuff. This is more for like fuel, if you're talking
Right.
about
Right.
starches
Oh,
and
we're
like,
we're gonna get to it. How
yeah,
sugars are
yeah.
actually, yeah. But for now, it's a little pointing at a little other nitty gritty fact about how they actually, store it, and how those two kingdom utilizes two types of starch, two different type of starch. And yeah, that's basically it. uh yeah, next slide. We have now the building blocks of DNA. So the where was I? Yeah. So last time we talked about the DNA pyrimidines and purines, but I did not have an image on the uracil pyrimidine. Here you see it, like how it differs from thymine, which is basically a methylated actually a demethylated version of timin. Um, yeah. So this is also to empathize on how the sugar type and phosphate group that composes our genetic information is assembled. So this is not DNA yet. It's what makes it work separately. So you have the bases, the sugars that's gonna link to the bases, and the phosphate group that's gonna link to the sugars. Any questions so far?
Uh, does the thymine become uracil when it's separated out? When you split the DNA into
Mm,
RNA? Or
I think there is a mechanism that actually does that. Although I would go for the RNA is assembled when the DNA is read in the transcription phase. We're gonna go through that in later courses how
Yeah,
the transcription works. But yeah,
yeah, that's an enzymatic process,
yeah,
and um, again, this is just about understanding that the things exist and kind of a overview.
yeah.
Um, uh, every single one of these questions are good questions, but they're questions that are like a week of normal school. So we have
Yeah.
to like build a foundation first here.
Of course, they're welcome, but just so you know, we won't elaborate too much on it. A little last thing is that the carbon in the sugars are enumerated right after the oxygen ketone in carboxyl. So you have like at the beginning of the carboxyl, the OH, H, and right after the ketone, the double linked O, you'll start with enumerating those as like 1, 2, 3, 4, 5, and yeah, that's it. We can go to next slide and see that this is energy. Adenosine triphosphate, known as basically the energy molecule for most life form, analogous to a charged battery. ATP will be used to release the energy that the most reactions in the cell depends on, and when that happens, you have ADP, adenosine diphosphate, which is the empty rechargeable battery that will be charged up again via phosphorylation. Any question on that? Cool. So the now that we covered most sugars on of our biological system, it's time to focus on what and how DNA itself is composed and which conformation it will adopt once created. On one side of the strain, it will act as a template, and on the other side, it will be a reading frame. As you can see, like they assemble like that. The illustration itself it talks for itself. So I almost don't need to say much if you just look at the images. So you have the sugar phosphate backbone, the hydrogen bonded base pairs that enters the double helix conformation. We're gonna cover that in the next slide. But any question before we go to next slide? Cool, cool. So at the next slide we have the the Baker pattern. Let's say on the top right, I took a little image from the manga guide to molecular biology, which is a cute version that says that uh, yeah. Uh,
I believe we have a
it,
copy in the library as well.
yeah. Okay. The minor and major groove is what helps us differentiate between these sense and antisense frame, like the 5' and 3'. They ensure a reasonable amount of stability, like the double helix formation, and the helix is formed by, like, yeah, as you see in the illustration, like if you would stack people on top of each other and tell people to hold each other's right or left feet, and then you twist those people, and if those people were like grouped in husband and bride, or like with their mates, they would automatically form a double helix. So yeah, that's kind of cute. Again, the illustration kind of speaks for itself. But I'd welcome any question here as well before we proceed. Okay. Uh, so methylation in DNA means silencing and preventing a specific sequence to be read. Whereas cytosine nucleotide is altered in the fifth carbon of the sugar, introducing a methyl group. The CPG side, also known as the CG side, are region of DNA where cytosine is followed by guanine. It basically means cytosine, phosphate, guanine, and those site occurs in mammals with high frequency. They usually are referred to as the CPG islands, and those are 70 to 80% methylated, which means like 70 to 80% of those sites are silenced. We only use very few genes that we're made of. And yeah, this example on gene regulation is called epigenetics. That's like, I don't know if you can feel it, but we're increasing the difficulty uh quite a lot now. Because the next courses are actually going to be molecular biology related, whereas this is still like kind of half biochemistry, half cell biology, and yeah, we're getting there, guys. Just buckle up and remember, it's only a few terms to look up, like away from getting a better understanding. So Wikipedia is your friend, the library is your friend, Google is your friend, or hell, DuckDuckGo or whatever search engine is your friend, Bing, or just use the internet. It's very useful.
Bing is not your friend. Don't lie to them.
No, nobody's your friend. Is You are your own friend, but yeah, just yeah. Who uses Bing? I don't know. I've never used Bing, but anyways. Yeah. Next slide. Or questions? Okay. Um. So the planar stacking is what helps stabilize the double helix. There is some numbers showing how tightly these molecules are packed together, and empathize how the orthogonal
I know you didn't. You don't need to. That's stupid.
Typhoon, would you mind
hello?
muting your mic? Thank you.
Okay. The planar stacking. So you have like an orthogonal and stacking. So you have like the orange rings and the green rings, which is like kind of what ensures the stability, like the major part of ensuring stability in DNA, and also a couple of numbers, as you see, like how far apart are those phosphor ions or phosphones. So you have like 0.34 or 0.34 nanometers, and once they're really stacked, you see the helix being like 2 nanometers in length. Yeah, it's pretty stacked, and it's gonna be even more stacked once we go to next slide, unless there is question. Okay, so before cell division, DNA is mostly spread out around the nucleus, allowing for certain regions to be read, and when the division happens, they all come together to form those chromosomes we often hear about. So the 10 micrometer to 1 micrometer difference in the picture may lead to the understanding that once they reach the chromosome site, they're actually packed into chromatin, and that is really like, yeah, that is kind of magic, but it's like how you would try to find a good analogy, but can't. I had one, but I cannot find it yet. But yeah, the image is quite cute. We know that when they divide, they all of a sudden become those very nicely arranged um, chromosomes, and yeah. Any question on that? Cool. We're good on time, so good. Next slide. Here is so here is the oof, yeah, all the different types of DNA sequence can be found in chromosomes. So you can see them like as beads on a string. So the higher order chromatin folding is then unpacked whenever they reach their destination, and you have loops of 30 nanometer fiber that are still on those beads of chromatin. Those beads of chromatin needs to be opened as well, or unpacked. With those mechanisms are going to be covered in the later courses. So it's basically to give you an intuition on it. It may look complicated, but we have been researching about those mechanisms, and even like the enzymes that unpacks tightly chromosomes are well known. So yeah, you have major type of DNA sequences: single copy gene family, tandemly repeated genes, introns. Introns, by the way, are those that those sequences that don't do much other than shuffling the exons, which are those that are actually going to code for proteins. And it's like a stack of card mentioned in the manga guide. It's by shuffling the cards, you can not only ensure that, or at least know that, okay, they do the same thing, but also so you kind of prove that no matter what happens, those genes are still pretty redundant and will still do the same thing. But also, in case mutation happens, transposable DNA elements, spacer DNA, simple sequence DNA, those types of DNA sequence, I am not going to go in details on those, but they are heavily studied. So those are rabbit holes you can get lost in or make a career out of, depending on what your mindset is. Any questions? Okay, so the next slide is gonna be a, a little peculiar per se. So back to reading chromosomes and pointing at two of the most important transcription factors from the Yamanaka cocktail. You have ep4 and SOX2 that basically usually tells the cells that they're in their embryonic state, that they're totipotent. Those are present in all the cells. They're just silenced. And yeah, methyltransferase initiate methylation or silencing. That means adding a methyl group, and the mechanism telling that cell needs to adapt that function will go in a chain, like in a chain reaction of silencing. So they won't just stop there and silence ep4 SOX2. They keep silencing uh, until they reach the like a site that tells them to basically stop. Uh, yeah. So that is silencing, for example, here totipotency, like to prevent uncontrolled growth and chaos within a system, aka cancer. Any questions? Cool. Sure. Now with translation, amino acids emerge from triplets or nucleic acid. There is no structural homology between different amino acids. It means like they all the 21 amino acids that we're made of don't look like each other in any way or have any. They all have a different purpose. Unlike, let's say, GC or AT, they all have like their specific functions. And how to say it? Yeah. The polarity of polymerization will be similar. Like each subunit of the chain decide what happened after and before it. So not only is each amino acid different in their function, like with their little things like the side chains, those usually carbon sticking out. It's also it's also like it's like typing a code on I can find many analogies, but it's like all programming analogy. I need to calm down with my programming. But yeah, that's basically it. Do you have any question on that? Okay. So with the circular version of the amino acid that encodes that are encoded with the three specific codons, the codon system. As you see, like let's take one that is like UGA. It is the opal stop codon, but in some organism you would actually make selenocysteine. So don't like whenever you see that, okay, this UGA or TGA has been making selenocysteine, you should know that it talks about another system. In our system, we know that those three TGA, TAA, and TAG are stop codons, and ATG or AUG is the initializing codon that tells, okay, we're gonna start translation here. We're gonna start making proteins here. Yeah. Questions on that? Okay. So converting mRNA. Next slide. Converting our mRNA is done in the ribosome, where transferred RNA dehydrates or metabolizes the next amino acids via the triplet decoding adapter, and it's a chain reaction, so to speak. Like it knows what to add before it's there because they follow a chain of it's only natural that the next one's gonna be that because they all read the same frame. Uh, Again, an illustration that speaks for itself. Oh shit! There you go. Okay. Yep. It's a lot of slides. It's also to make it very visual instead of me talking all the time. And yeah.
Is there a difference between RNA and mRNA?
Yeah, as in like you have the transfer RNA, which is an RNA that is already made for carrying the amino acids to the site where they will be added together, polymerized, or yeah, insert name of putting things together. Yeah, man, I went through it fast this time. Nice. Okay.
Next slide.
Yep. Transient transfection means that the constructed or transcribed plasmid are introduced into cell in some ways without integrating foreign genes into the genome of the organism. During growth and division, these transfected genes will be gradually lost, and is also why mitochondria and chloroplasts are replaced often to ensure correct ATP activity and replicability. Like they have plasmids. Like we in our mitochondria we have plasmids, and those plasmids are usually lasting. Forgot the numbers, but they don't last long. So after a couple of cycles, they even if the even if the mitochondria or the chloroplast has not been used much for ATP, or if the plasmid are still intact, there is a mechanism that says, okay, you're gonna be ligated, because we cannot make sure that you're gonna do the same thing in the after the fifth generation. It's usually 5 cycles and then recycle. So yeah, here we're nearing the mechanism of protein expression, but this slide tells a lot of things that you may want to ask any question on. Like, are there any, for example, electroporation, or are there anything you want to know that I may or may not explain in detail? But no. Okay. So last slide. Examples of the structure and assembling on each domain has been picked to display a vast field of multidisciplinary science within, or in what we call biology. So biology is super multidisciplinary, from computer programming to physics, you know, chemistry, mathematics. Like it's a lot of people. It takes a lot of people to even understand those phenomena. And I have skipped many steps in both the cell biology and biochemistry's section, because we simply don't have time. We don't have the qualification, nor are we. We cannot just make a whole course that will explain most of the things, because we don't have countless amount of semesters. So what I'm doing is basically pointing at different things, even things that I'm not myself completely sure about, but it's basically to tell you, hey, those are the things. Like they may be rabbit holes, you may get lost in them, but they're good rabbit holes, and they're very useful, and you may even make a career out of it. That's basically it. So I can only advise you to make the effort of looking up those terms, that they're readily available on Wikipedia. Hell, like if you see my bot where you say exclamation mark TR, you insert and that DNA sequence, and it will translate it. Like that was made from looking up Wikipedia pages. I didn't even need textbook for that. Like it's on Wikipedia, like the information, not the source code for the program, but like let's say the anaerobic and aerobic cost for making those proteins, or their masses, their monoisotopic masses, or and I have so many more. Like you have the block substitution matrix, the PAM. Like you have so many things that I actually I'm thinking of making more than one function, more than exclamation mark TR. So like a simple translation, then useful translation for the ATP cost, because it's important to know if you have a yeast that's gonna make insulin, you kind of want to know how much energy it's gonna need, and thereafter you're gonna insert X amount of ATP constituents, so that it doesn't run out of energy, or it doesn't have an excess of each type, for example. And also like yeah, uh, I'm not talking about the slide itself. As you can see, it's and that's for a specific purpose. So when you look at this simplified plasmid map, I assume you can already tell what some of the elements are, right? I mean, from what we've talked about earlier on, you may already know that the inserted gene from Justin's video or 5', 3', a primer or antibiotic resistance genes, like those are elements from either journal club, like they're all around you. So it's just to make you feel like we're almost there, guys. Like you are way closer to understand how a plasmid worked than you were before. So it's, yeah, congratulations and thank you for your time. I mean that's it. That was the end of course 2.
Anyone got
Anything?
questions?
Yeah, thoughts, comments?
There's a lot of information thrown out, so someone must have questions, surely.
Or maybe they're just overwhelmed. Yeah, I may have gone a bit too quickly through it.
I'm gonna reserve questions for my second or third go through on this one.
Okay, fair enough. I mean, I'm always there on Discord if you need to ping me for anything. But if it is something that you can look up on Wikipedia, I might do the search for you and give you the link, or I might tell you it's the first search result. But if it's not, and if it's something I know, I'll definitely tell you about it. I'll do what I can, guys. But I'll never talk down on you. Like even if it's like an obvious, if it has an obvious response, I won't just say, hey, this is a stupid question. Like I'm
Well,
not like that.
you don't know what you don't know. That's the thing.
Right, exactly. Okay, guys.